Distinguished Pro-Vice-Chancellor, Provosts of colleges, Deans and Directors, Heads of Department, members of the leading team of the Young Researchers Forum of the Office of Grants and Research of the Kwame Nkrumah University of Science and Technology, ladies and gentlemen, I bring you greetings from the Institute of Distance Learning Conference Center of the Kwame Nkrumah University of Science and Technology, where we are hosting the maiden edition of the renowned Scholars Dialogue. My name is Marian Asantua Nkansa, your moderator for this event. And as we await a befitting welcome from University Management, let me take this opportunity to welcome you all to this space, especially those joining us virtually on YouTube and also via Zoom. The essence of this dialogue series is to feature annually an eminent person who has walked the path of research and has golden experiences to share. We are excited to be hosting a Nobel Prize winner for this inaugural edition. For an extraordinary event like this, we need an extraordinary person to oversee affairs to a successful end. The person to help us do this is part of university management. He is a physician by training and a public health expert. He lives, breathes, and eats research, and has the welfare of the Young Researchers Forum very dear to his heart. Ladies and gentlemen, Professor Ellis Owusu-Dabo, the Pro-Vice-Chancellor of our university, is here to chair affairs on behalf of the Vice-Chancellor. Thank you, Prof, for coming.
We need to dream of science and technology. We believe that beyond living the dream of science and technology, there's a need to create what we refer to as research citizenship. That will create the enabling environment to ensure that all students and faculty understand and know that one of the principal ways of transforming our economy is to showcase the evidence that we have and through the research that we conduct and bring about the tangibilities of those research described as technology. It's important that whilst we're pursuing this agenda, that we look at who will do this. And certainly, we don't just look at the present generation, but we also look at the next generation. In line with this, the Vice-Chancellor, when she was appointed sometime in 2020 in August, as part of her vision, was to introduce what we refer to as Young Researchers Forum, at which forum we will bring researchers together. I still consider myself young, so I'm part of the Young Researchers Forum. Bring researchers together so that we can have a dialogue and create a certain momentum that allows not just the present generation, but the next generation scientists to be able to nurture research. As part of the activities of this forum, the lead, Professor Marian Nkansa, and team have what they refer to as the renowned Scholars Dialogue. And what a way to begin this one. I have had the opportunity of meeting with once in Massachusetts, meeting with a Nobel Peace Laureate. And it's humbling when you meet with such individuals who have accomplished to be able to engage with them, listen to their thinking, listen to where they have been and how they've made it, and what lessons we as young researchers can pick up from there. Today, we're privileged to have one of the renowned Nobel Peace Laureates in 1993, Sir Richard John Roberts, who will talk to us in this conversation. I want to say that this is not only a dream come true for us, but importantly, it is an inspirational gathering time for us. And I'm hoping that those of you who have joined us from far and near via the virtual space, you pick up your notebook and take profuse notes, but importantly, engage with Sir Richard Roberts, ask him the questions that you have to ask, and importantly, pick up the lessons that have brought him where he got to almost 20, is it 30? Almost 30 years ago. Almost 30 years ago. So, colleagues, friends, this is a rare opportunity. And I'm hoping that we'll seize this opportunity and make it count and pick up important lessons that will take us to where, hopefully, as a university, we want to get to. It is not so much as about our visibility as a university, but it is so much as in the belief and the practice of research and research management, and creating ultimately a research citizenship to which every faculty student believes that the way to transform our economy is to churn out the evidence, believe in the evidence, practice the evidence that we churn, and hopefully encourage politicians or the policymaker to also pick up this on board. In that way, we will be able to transform our lot and ensure that we get to our destination of true academic freedoms propelled by research citizenship. And on that note, and without wasting any more of your time, because the conversation is what we are all looking out for. I can see you, Sir Roberts, and we're hoping that we will have a fruitful time with you, engaged, and a memorable one, of course, going forward. I thank you all for coming on behalf of management. Vice Chancellor is out of town, would have loved to be here for sure, but an equally important reason has taken her out of town. And I'm hoping that all of you from the virtual space and those of you here will enjoy this momentous occasion. I thank you so very much, and God bless us all.
Thank you very much, Professor Ellis Owusu-Dabo, for these warm remarks that you have shared with us. Next on our schedule is remarks from the Director of the Office of Grants and Research. The Young Researchers Forum works directly under the Office of Grants and Research, supporting their efforts in achieving Theme 2 of the University Strategic Goals. Ladies and gentlemen, Professor Philip Ncheje.
Good afternoon, everyone. The Pro Vice Chancellor of the University, Professor Ellis Owusu-Dabo, who is representing the Vice Chancellor this afternoon, and who is also chairing this meeting for us. Provost of the colleges, my own provost, Professor Mekuji, Professor Ajari, deans of the faculties, various faculties of the university, heads of departments, ladies and gentlemen. And let me also say the executive of the Young Researchers Forum. I was the lead or the chair for the Young Researchers Forum until I was asked to direct affairs at OGR. The Office of Grants and Research supports research enterprise throughout the university, providing support to all the 6 colleges of the university. We make sure that grants are disbursed according to university regulations, according to national and international standards. We do this by also making sure that we provide the necessary training to our researchers and also making sure that we look for opportunities for funding for them, irrespective of the particular discipline that you find yourself in. But I think, as the Vice Chancellor mentioned, the Young Researchers Forum was established last year under the Office of Grants and Research to sort of provide a platform to engage our young and mid-career researchers, including myself, and as the Vice Chancellor said, including himself. In Ghana, up to 40, 45, you can still count yourself to be a younger person here. So I'm sure many of us here can be considered to be young and therefore be part of the Young Researchers Forum. I have had this question that has been thrown to me in a number of occasions: who qualifies to be part of the Young Researchers Forum? And I think as a committee, we have often debated on who qualifies. For me, anybody who wants to be part of the research enterprise in the university is welcome. Whether you have even if you have retired and you want to be part of it, our doors are open to you. The university seeks to become a research-intensive university, and I've always said that we can only do that if we try to change the kind of paradigm that we have and change into making sure that research becomes part and parcel of everything that we do here. Our teaching cannot be meaningful if you don't engage in relevant cutting-edge research. And for that matter, I hope that all of us, those who have joined us online, especially the young guys, the young ones who have joined us recently, who have been appointed recently, you join the Young Researchers Forum. We share opportunities. Sometimes we are not able to share across all the networks. It takes time, but as soon as we get something, we put it there for people to apply. This afternoon, as the chairman said, we are blessed to have one of the finest. I'm sure if it were to be in the among the football fraternity, maybe we'll be calling him Pele of Brazil, or something, somebody of that nature. So this afternoon, let us be attentive, listening to him, what he has to share with us. Let's take some notes so that when we leave here, we can digest, we can reflect on some of the things that he'll be sharing this afternoon. On that note, on behalf of my team at OGR, I think the Vice Chancellor has already welcomed all of you, but we do wish you a fruitful discussion, and we say God richly bless us all. Thank you.
Thank you very much, Prof, for being so gracious with your words and your time spent with us. For those joining us virtually, please use the chat to introduce yourselves and feel welcome as well. So you tell us who you are, your name, and then where you are joining us from all over the world, I guess. A wise man once said that the greatest gift you can give to someone is your time, because when you give your time, you are giving a portion of your life that you will never get back. Our speaker today has been gracious with his time and will be sharing a piece of himself, if not all of himself, with us. Sir Richard Roberts is an entrepreneur, molecular biologist, and architect of gene splicing. He's a chief scientific officer for New England Biolabs, headquartered in Ipswich, Massachusetts, in the United States of America, where he has been a director of research since 1992. Sir Richard's laboratory has a long history of research on restriction enzymes and their associated DNA methyl transferases. He is involved in developing methods to find new enzymes with novel properties and using a combination of bioinformatics and biochemical experimentation to probe DNA sequencing information from the many microbial genome sequences that are now available. Sir Richard's research focuses on finding general ways to predict function in such cases and to use bioinformatics to make predictions that can be tested experimentally. He has authored over 300 publications and more than 10 patents. He has served on a number of scientific advisory boards, including Celera Genomics, Genex, Oxford Molecular Group, and Lennox Biotech. Sir Richard Roberts was awarded the Nobel Prize in Medicine and Physiology in 1993 for the discovery of introns in eukaryotic DNA and the mechanism of gene splicing. He shared this Nobel Prize with Philip Sharp of MIT. Since winning the Nobel Prize, Sir Richard has been involved in organizing a number of Nobel initiatives to correct scientific misunderstandings and promote humanitarian causes. His most recent campaign has been on the issue of GMOs, where 158 Nobel laureates have supported the use of GMOs techniques to improve plant breeding practices that could greatly help developing world. And most recent of his endeavors was his quest for positive social change and impact, which has led to an appeal to co-organize with Prof. Hoffman, Nobel Prize winner in chemistry for 1981, to address the conflict in Ukraine. This appeal so far has been signed by 165 Nobel laureates. Ladies and gentlemen, Sir Richard Roberts.
Okay, thank you very much for the introduction. I trust you can hear me now.
Very well, sir.
Okay, I will. Okay, can you just confirm that you can see that properly?
Very clearly.
Okay, so we need to go back to the very start. Okay, so what I want to do today is to give you a quick run through on my life, starting as a very young boy, how I got interested in science, how that translated into a career in science, and eventually how I made the discovery that led to the Nobel Prize. My aim in this talk is really to give you that made a difference. And I think you will discover as we go through the talk that the most important thing that happened to me is called luck. I have been incredibly lucky throughout my life. But I would also note that everybody has luck. And the most important thing about having luck is that when you have it, take advantage of it. Don't pretend that it was something that just happened to you and never happens to anybody else. It happens to everyone. And the very first lucky thing that happened to 43 in Derby, England, it's in the center of the country. And I was living there almost towards the end of the Second World War. And something very lucky happened. It turned out the street I was living on was on the direct flight route from Germany to the Rolls-Royce factories that were making the engines for the RAF bombers. And the Germans were intent on bombing that particular factory. However, the bombing accuracy was not so good, and they dropped a bomb on the street on which I was living. But fortunately, the bomb landed on one side of the street and I was living on the other. Very, very lucky. I was going to be able to take advantage of, but I think it just points out to you that you never know when luck is going to play a part. Now, in 1947, when I was just 4 years old, my parents moved to Bath in England. This is down in the southwest, very close to Bristol. It's about 100 miles due west of London. Very beautiful city. And I really grew up in Bath. I don't have much recollection of anything that happened when I was in Derby. Now, the school that I went to, the junior school's called St. Stephen's School, and something very lucky happened to me there. When I was doing exams and so on, and you know, just learning how to function in society, the headmaster must have seen something in me. I don't know what it was, but he used to give me puzzles to do after school. Not every day, but very often. And they were mostly mathematical puzzles. And I just loved them. I fell in love with mathematics at the time. And I only later discovered that he wasn't doing this to everybody. He was just doing it to me. His name was Mr. Boakes. And I'm very happy that he learned that I had won the Nobel Prize shortly before he died, because he played a very important part in my early days. And I didn't even realize it, that he was getting my mind thinking about mathematics and really thinking scientifically, as opposed to thinking about. By age 11, I decided I was going to become a detective. I'd read a book entitled How I Became a Detective, a regular detective in the police force. And I really felt that this was what I would be good at. I love solving puzzles. I had a great curiosity. And of course, being a detective is simply solving puzzles, but in the location of a crime. I show you the two pictures here because they illustrate something very nice about the city of Bath. At the bottom left are the Roman baths. The Romans had discovered this city, and they built these beautiful baths around some natural hot springs that were there. And the top right is the Royal Crescent, which reflects the history of Bath in the Georgian period, when they put up very beautiful buildings. Now, when I was about 11, my father gave me a Christmas gift, and that Christmas gift was one of these chemistry sets that you buy, and it had a lot of interesting experiments you could do, and it had some chemicals. And in those days, the chemicals that they gave with the kit were much more interesting than the ones you get today. And in fact, I discovered that I could make fireworks and I could make explosives with the things that were in this chemistry kit. And as soon as I was making fireworks and learned how to blow things up, I thought this was great. And I really fell in love with chemistry and decided that rather than just doing math, I would do chemistry. Now, at age 11 in England at the time, you had to take an exam called the 11+ exam, which was to tell you whether you were suited for a more academic career or for a more technical career. And it was interesting, in those days, I took this exam without even realizing, none of us in the class realized it. We were just taken to class one day and given an exam, and we all sat there and did it. And it turned out it was this 11+ exam, which was going to determine the future for you. No special preparation, no extreme, shall I say, for it. We just took it. And I did extremely well in it. In fact, I learned very much later that I had received the highest score in the whole city for mathematics. And so that was good, but I got to grammar school. And the grammar school that I went to was at the time called the City of Bath Boys School. It was sitting up on a hill on the city of Bath. And in order to get to school, I lived on the other side of town. I had to walk down to the river and then walk back up the hill. And so I ended up walking uphill both ways in order to go to school. And while I was there, I increased my love for mathematics. I thought this was great. We did some chemistry and some physics. I really hated physics. Was just not a good teacher. I also learned how to play the violin and joined the school orchestra, and that was a lot of fun because the school orchestra gave concerts and went off on trips to places. But one of the things that really was important for me was that I discovered caving, that is going underground. Very close to Bath is a limestone escarpment of the Mendip Hills, Cheddar Gorge. You may have heard of Cheddar Cheese. It's very famous cave there, but the whole area is just full of caves. And so I got interested in caving, and that turned out to be a very important activity for me because I didn't like any of the traditional activities of sort of playing cricket and playing football and rugby and so on, the sort of things that one normally does in school by way of sports. Unfortunately, a teacher came along who was also a big caver and persuaded the school that caving could be an acceptable activity to replace cricket and rugby and so on. And so that was very nice. And I also discovered jazz, for which Bath became quite famous. They have a very good jazz festival every year, and I enjoyed that very much. And between playing the violin and classical music and jazz, I had a much broader love of music. Now, in 1962, I went to Sheffield University, and I got there, again, a little by chance. I wanted to study chemistry, and there were two really good universities that did chemistry. One was Southampton, which was just south of where I lived in Bath, and the other was Sheffield in the middle of the country. I applied to both. I got accepted to Sheffield, but I didn't get accepted to Southampton. So there was no choice as to where I was going to go. I went to Sheffield, and that turned out to be another very fortunate experience. I had a great time there. I learned several things, one of which was how to play bridge, the game of bridge. And that occupied a considerable amount of time when I wasn't studying. And it was quite often that I wasn't studying, so I played a lot of bridge. My interest in chemistry, plus this fascination with games and puzzles, really led me to pursue a career in research, because it was a chance to be a detective and solve chemical puzzles in the world of science. I got my degree, my first degree in 1965, and then went on to do a PhD. And I was extremely fortunate when I was doing the PhD because I was put in a lab where there was a Japanese postdoc called Kazu Kurosawa, who played an important part in my life. He really taught me how to do chemistry in a practical sense. He was one of these wonderful teachers who would tell you, well, you should do this and this and this, and would explain why you had to do it. And so as a result of that, you did an experiment. Most of the time it would work, but you understood why you had done it and why it worked. And that was really excellent. I did my PhD with W.D. Ollis, David Ollis, and he had made chemistry, organic chemistry, he was professor of organic chemistry, had made it fun. And he'd made it fun by emphasizing the use of problem solving when he was teaching. So the way he taught organic chemistry was to give you problems rather than to just recite facts and say, well, memorize this and say, remember this and this and this. And he really made it fun. I've never had as much fun in a class as I had with him. And so that was really very nice. And Sheffield is also an interesting university. They have a number of Nobel laureates associated with them, shown here. But it turned out that I was their first student to win a Nobel Prize. And that I thought was pretty good, but they thought it was fantastic. And they made a huge fuss of me. But then a few years later, Harry Kroto, shown on the bottom right, also a chemist, won the Nobel Prize in chemistry And he too had been a student at Sheffield University, but he was ahead of me. He was 4 years ahead of me. But he always used to like to tell people, because he was older, that he taught me everything I knew. In fact, we never met when I was at Sheffield, but I would always respond and tell. It took him an extra 3 years to win his Nobel Prize. So it was a fun experience. We had a lot of fun. Harry was a great lecturer and also a very, very good teacher. He was very entertaining for students. He would have done wonderful at an event like this. I also should correct one small point from the introduction. My Nobel Prize was not in peace, it was for medicine or physiology. Now, when I was a student, I actually, thanks to Kazu, I had pretty much all of the results that I needed for my PhD by the time I'd finished my first year. But in England, you had to spend twice. And so for the next 2 years, the first year I sort of did some experiments that I had thought of that didn't really work out terribly well. But I also spent a lot of time in the library. And I would read a lot about subjects other than chemistry. And this is something else I would say to all young people. It's often tempting just to focus on whatever subject you're trying to learn. But in fact, it makes much sense to read about other things, sometimes things not even terribly related to what you're doing. Because sometimes when you're doing that, you discover that something else is far more interesting and far more fun than what you're studying. And if that turns out to be the case, I always recommend changing your career. As I told you, when I was very young, I thought I was going to be a traditional detective, and then I discovered chemistry, and so I moved into chemistry. Well, when I was a chemist, I read this book by John Kendrew. It was called The Thread of Life, An Introduction to Molecular Biology, and it was about the origins of molecular biology and where it came from, about crystallography and about DNA eventually, and so on. And this, I was absolutely captivated by this. I thought this was wonderful. And by the time I finished the book, I knew that this was what I wanted to do with my life. I wanted to become a molecular biologist. So I wanted to switch from chemistry. And it's always good timing if you can make these kinds of career decisions when you're switching from one job to another. So after finishing my PhD, I had to do a postdoc. If you wanted to get a job in England, you really needed to go abroad and get a postdoc. And so I applied to half a dozen labs that were molecular biology labs, hoping that one would take me and accept that, I was there to learn molecular biology and use my chemical background and my chemical training to do so. And so I applied to, 6 places. Only one person accepted me, and that was a man called Jack Strong. And I thought I'd be going to Wisconsin, and then 3 months before I was scheduled to start my postdoc, he wrote to me and he said, well, could you just delay for a little while because I'm moving to Harvard? And so quite by chance, again, a stroke of immense luck, I ended up at Harvard instead of the University of Wisconsin. And that turned out to be good for many reasons. But one reason was that Jack Strominger had just achieved his lifelong ambition. He'd always wanted to be a professor at Harvard. He just achieved it. And so whereas he had a reputation for working his students very hard and really bearing down on them, he took this opportunity to relax a little and to change fields. And I ended up, working for this gentleman, Jack Strominger. And while I was there, I picked up on a project that he had started several years previously. He'd been interested and had made his career working out how bacteria made their cell walls. And one of the interesting features of the cell walls of many bacteria is that they have cross-links of amino acid chains that kind of hold the peptides together and the proteins together that form the cell wall. And one of his previous postdocs had found that there was a glycyl tRNA that was responsible for transferring the glycine into the cell wall. And this particular tRNA did not work in protein synthesis. It only worked in cell wall biosynthesis. And so it was interesting to determine what the sequence was and find out why it couldn't function in protein synthesis. And so this was the problem I was given. Now, the postdoc who I overlapped with for a few months had started on what we call a traditional way of determining sequences, which was one developed by Robert Holley, that involved making large amounts of tRNA and then cutting it into pieces and doing a lot of more traditional chemistry on it. But I started to read the literature and discovered that a man called Fred Sanger had actually developed a better method for sequencing RNAs. This one's involved making P32 labeled RNA and then using a lot of fairly advanced techniques in order to sequence it. And so as soon as the first postdoc left, I switched immediately and started making some P32 labeled tRNA and then applied to Fred Sanger's lab in order to sequence it and to learn the methods of sequencing. Now, Fred Sanger was a remarkable man. He was someone who had first won the Nobel Prize for developing techniques for sequencing proteins. He then went on and determined methods for sequencing RNA and later developed methods for sequencing DNA. And he shared that prize with Wally Gilbert, although it was really Fred Sanger who made the key advances in this field. And his method was far superior to the one that Wally Gilbert developed. Anyway, in 1969, I visited Fred's lab to learn how to do RNA sequencing. I spent 6 weeks there, came back to Harvard, and Jack Strominger was good enough to provide the money to set up a lab that would emulate the lab that Fred Sanger had at the MRC lab in Cambridge. And so I started sequencing RNA, and I was the first person in the Boston area, it turned out, to be sequencing RNA using Sanger's techniques. And as a result of that, everybody came to me to learn how to do it, and including Wally Gilbert's technician, Alan Maxam. He came to learn how to do it. In time, I made a lot of friends. I got a first author Nature paper, well, actually a sole author Nature paper. Jack Strominger didn't want to put his name on it because he said he'd done nothing to help. And so I ended up with this first author paper, only sole author paper. Anyway, so that was good. So I had a very nice time at Harvard. It turned out I met so many fun people, interesting people, and people that in many cases, I've kept in touch with for the rest of my life. Now, one of the things that happened while I was there was that one day, Mark Potashny of Lambda Repressor fame came and said that Jim Watson was looking for someone to go to Cold Spring Harbor to sequence SV40 DNA. And he said, I told Jim about you, and he will come and talk to you about this. And so I waited, and a couple of weeks later, Mark came to me and he said, well, Jim doesn't actually know who you are, so perhaps you could go and introduce yourself. And so I did. I went and introduced myself to Jim, walked into his office, told him who I was, and within 10 minutes, he offered me a job at Cold Spring Harbor Laboratory. I ended up accepting the position, although I'd actually been also offered a job at Harvard in the biology department there. But Cold Spring Harbor sounded more interesting. And of course, at the time, Jim Watson sounded like a more interesting figure. Anyway, so I go to Cold Spring Harbor and I discover while I'm getting ready to go down that there are two other groups who are already sequencing SV40 DNA. And I've never really liked too much competition when I do my research. I like to work in fields where there are not a lot of people working. And so I decided that rather than just sequence SV40 using the techniques, the RNA techniques, basically the way people were doing it, there were no techniques to sequence DNA directly. Everybody was making copies of it and then sequencing the RNA and putting it back together. And I thought, well, maybe there's a way that one could sequence the DNA directly. And this came as a result of listening to a lecture by Dan Nathans. Dan Nathans, who went on to win a Nobel Prize a few years later. But he talked about this enzyme called endonuclease R, which was a restriction enzyme that would cut DNA into very specific pieces. It occurred to me that if there were more of these, then we might be able to go ahead and sequence DNA based upon the recognition sequences of these restriction enzymes, but also they offered a way to make small pieces of DNA, which was what you needed in order to develop new techniques. So I went to Cold Spring Harbor. I immediately started making endonuclease R, and at the time there were 4 other restriction enzymes known. I made those and we started to look at them. And then I brought with me a bunch of bacteria that I'd saved from Jack Strominger's lab. And so I started opening those up, and it turned out they all had restriction enzymes in them too, and they were different from the ones already known. And so during the 1970s, from 72 onwards, and the early 80s, 75 of the 100 known enzymes at the time were isolated in my lab at Cold Spring Harbor. And these were the things that really got the biotech revolution going, because they offered you a way to cut out genes and then move them from one place to another, or to sequence them, or whatever you wanted to do with them. But there were just no other tools that would allow you to do what the restriction enzymes would do. And everybody was coming to me and saying, oh, could I get a little bit of this? People would come to meetings with tubes of DNA in their hands. And they say, oh, could we just see if you have a restriction enzyme that will cut it? And so my technician had set up a little panel that had all of the known restriction enzymes at the time. And then these people would come and just take a pipette, take out a little bit and see if it would cut their DNA. And it was obvious there was a business here, because I wasn't going to supply the world. And I said, look, why don't we set up a company to make these enzymes, and we'll use the profits to support the research at Cold Spring Harbor. We can set up the company somewhere else, but then just use the profits to support research at Cold Spring Harbor. To which his response was, well, oh, that's silly. He said, you can't do that. You're not going to make any money doing that. And he had no interest, didn't want to do it. But I knew that there was a company that could be made from this. And so I started to ask around to see if there was someone else who would be interested, or to see if there was a company that might be interested. And I ran into a man called Don Combe, who had started a very small company in Beverly, Massachusetts. And he too had had this idea that restriction enzymes might be interesting. And he had proposed to sell just 2 of them through a company called Miles Laboratory. And in fact, it was Miles who put me in touch with him. And at the time, Don Combe, the company now called New England Biolabs, at the time it was just called Biolabs, was composed of 3 people. It was him, his wife, and 1 technician. And I'll tell you a little more about that later, because that is a sideline from the main story leading to the Nobel Prize discovery. These were the men who won the Nobel Prize for discovering restriction enzymes. On the left, Werner Aber discovered the very first type 1 restriction enzyme, which turned out not to be useful as a tool, but was a very important component of the way in which bacteria defend themselves against bacteriophages. In the middle is Ham Smith, who discovered the first type 2 restriction enzyme, the one that was called endonuclease R from the previous slide. And he had discovered this enzyme because he was looking for methods of transduction in bacteria And he was interested in the enzymes that might allow DNA to move from one bacterium cell to another. He had no idea what this might be useful for, but Dan Nathans, the man on the right, the man whose lecture I listened to and convinced me that restriction enzymes had much better uses, he had figured out that they were very good for mapping genes in SV40 DNA. And so there was a connection with SV40, there was a connection with restriction enzymes. Now, I have to say, Jim Watson was totally opposed to all of this. He said, in fact, he told me he'd hired me to sequence SV40 DNA, and he thought I was wasting my time with all these restriction enzymes. But I was able to convince him, I guess, that I was doing something that might look as though it was helping to sequence SV40 DNA. So this was really how I got started in restriction enzymes, and they've been a passion of mine from that day on. So this is a little bit about what happened with restriction enzymes. So between 72 and 75, we discovered 30 restriction enzymes, all of which had different recognition sequences. And the reason that this was possible was because almost all bacteria have these enzymes. There are many different specificities. We now know of hundreds of different specificities. And so if you start to look in different bacteria, you quickly found different sequences being recognized. So that was very good. So in 1975, I became the official scientific advisor to New England Biolabs. We gave them the strains from which we'd isolated the enzymes. We told them how to make the enzymes, and they started to sell them. I should say at the time, we didn't patent any of these. And in fact, these strains that produced them, I sent around the world. They went to Japan, to Germany, everywhere, to people who wanted to make the enzymes, including many labs that wanted to make them themselves. In 1975, I started a list, which ended up being a database that I run today, now called Rebase, which keeps track of all of the restriction enzymes and restriction enzyme components that are present in bacteria and in archaea. These enzymes only appear in bacteria and archaea, they're not in eukaryotic cells. In 1976, many genes for restriction enzymes were cloned at New England Biolabs. And in 1978, my lab started working on bioinformatics, and we in fact wrote many of the original programs that were used to assemble DNA sequences from small fragments and to start to analyze them, looking for genes, looking for protein coding regions, and so on. Now, this is where we got started on the work that led to the discovery of RNA splicing. So some of the restriction enzymes were being used to map adenovirus DNA, and I had started working on adenovirus DNA because I lived on the lab grounds at Cold Spring Harbor, and underneath me in the apartment below was a Swedish gentleman, Ulf Pettersson, and he had bought adenovirus to Cold Spring Harbor. This is a eukaryotic virus and a very good exemplar of what eukaryotics do. If you want to study eukaryotes and genes and mechanisms and so on, this was a good way to do it. And so in 1974, my postdoc, Richard Gelinas, I gave him the project to start working with adenovirus 2 messenger RNA. And the idea was a very simple one. We really just wanted to know if the promoter region in a eukaryotic cell was the same as a promoter in a bacterial cell. The promoter is the sequence that precedes the start of messenger RNA production. And so there was a question, is it the same? Does the RNA polymerase recognize the same sequence in bacteria and in eukaryotes? So to cut a long story short, we started, I came up with what I personally thought was a very clever method to get at the sequences of the promoter. However, it turned out that things didn't work out quite the way that I expected. And so I learned something really important then, which again is something that's stayed with me for my entire life. And that is, we did this clever experiment and it didn't work. And so failure can sometimes be a really good thing, because when something fails, it's usually because you did something wrong, or in the case of science, because nature is trying to tell you something, but what you thought was true is not in fact true. And that was the case here. And so we started to work to figure out why these experiments were failing, and we ended up discovering RNA splicing. We spent about a year really trying to find ways of proving what was going on, of showing what was going on. And we had ideas about, how it might be all working. But what we knew was that the late messenger RNAs, of which there were about 8 to 10 in adenovirus, they all had the same sequence at their 5' end. And so the question was, how did it get there? Well, one Saturday morning, I devised an electron microscope experiment that I thought would tell us. Now, neither Richard nor I had any experience or expertise in electron microscopy, but fortunately, just down the hall, there were some molecular biologists, some electron microscopists rather, who did. And I'll come on to them at the moment. So anyway, we started working this. This is Richard Gelinas, the postdoc who did all of this work. He has now retired and is living over in Washington State, near Seattle. And this was the paper that came out, which said that there was this one 11 nucleotide long T1 fragment at the end of adenovirus messenger RNAs. They all started with exactly this same sequence. Now, Tom Broker and Louise Chow were the electron microscopists who were working downstairs. We went to them on a Saturday morning, and this was when I can you do this experiment for us? We'll make the reagents that you need, but do you think you could do it? And they looked at the thing and they said, well, no one's ever quite done that before, but it should be doable. And they didn't see why not. Richard worked on Sunday and Monday to make the RNA components and the DNA that we needed for this experiment. And on Tuesday morning, Louise Chow, who was married to Tom Broker, came and told us that, well, they'd done the experiment, that the answer looked exactly like the picture that I'd drawn on the blackboard on the Saturday. And at that point, RNA splicing was discovered. Because I'm sure you all know, certainly any of you who do biochemistry, that to see a picture of something is always very, very much informative and convinces people more so than a set of complicated reasoning based on biochemistry. And so here was a picture of what the thing was looking like and what it should be. And so that turned out to be really good. By the end of that Tuesday, people were already talking about Nobel Prizes. This was in 1977, because it just threw on its head everything we thought we knew about eukaryotic DNA. It showed that the genes were split into pieces that needed to be joined together in order to function. We started talking about this, we told many people, and in very short order, a large number of other labs did exactly the same experiment. There was one really nice thing that happened at a meeting of RNA tumor virus meeting at Cold Spring Harbor in May. I came up and gave a little talk about this discovery beforehand, and Mike Bishop, who was one of the speakers at the meeting, later showed his picture, which showed that splicing was taking place. He had was by RNA splicing. And so he was able to immediately talk about this in his talk because he'd heard about this. Prior to that, he confessed he had no idea what was going on. And so that was very rewarding too. But of course, it also led to a lot of competition. Many people rushed to try and get a 1977 publication date. And they all like to claim that they had discovered it first. In many cases, this was because they had done their experiments in the genomes of eukaryotes, in yeast, or they'd done it in plants, or they'd done it in humans. And they all try to claim, well, we just did it in viruses and it wasn't quite the same. And what was really important was what was in eukaryotes. As a result of that, it took quite a while before the Nobel Committee came around and decided that the work that I did and the work that Phil Sharp had done independently at MIT at about the same time was worthy of the Nobel Prize. And so that was very good. So this was really a wonderful discovery. We tried to follow doing some experiments to see if we could recapitulate this in vitro. Could we set up an in vitro system that would work? And we spent several years trying to do this. And the idea was to make RNA in vitro and see how it could get spliced. And we did this using a bacterial promoter to make a eukaryotic gene, to copy a eukaryotic gene. And we used a polymerase from E. coli. And it turned out that that was not the way to do it. And Tom Maniatis at Harvard independently set up a system in which he used a much more specific RNA polymerase and promoter and was able to follow all of that in vitro and to get RNA splicing working in vitro. So we got scooped on that, which was fine. That was, that's how science is. But shortly thereafter, I decided that so many people were starting to work in this field that I didn't really want to work on it anymore. And so we started to concentrate on bioinformatics and sequencing. We sequenced the adenovirus genome, which at the time was the second longest sequence that had ever been done, and went on and developed bioinformatic methods and did a bunch of stuff there. But I want to divert this point and tell you a little about New England Biolabs. So as I say, New England Biolabs actually started out as Biolabs in 1974, switched the name in 1975, and I became their chief advisor until 1992. And in 1992, they had moved from a basement in Beverly underneath a hairdressing salon where they got started into this nice building shown on the left-hand side, a beautiful building that had been put up. And because they made a lot of money selling restriction enzymes and other things. In 2005, the company moved to Ipswich. Now, of course, 1992 was 1 year before I won the Nobel Prize. And so I hadn't been there very long when a whole new career really opened up for me. So let me just tell you a little bit about what went on at Biolabs until, So this slide is what we've been doing at Biolabs, what I've been doing since I became a research director there. So we did a lot of cloning of restriction enzyme genes for commercial production and other genes too, RNA polymerases and various other things. We did a lot of work on bioinformatics. Number of people at Biolabs doing bioinformatics. And then we did a lot of genome sequencing with the idea of using the genome sequencers and some fancy software in order to find the genes that encoded restriction enzymes, see if we could predict specificities, see if we could find ones that were new that would be of use to the academic community. But then, of course, I was awarded the Nobel Prize in 1993. I shared it with Phil Sharp, shown here on the left, and a much younger version of myself shown on the right. And I have to say, this was obviously the highlight of my life, as you would expect. It was really a wonderful time. We had a very interesting trip to Stockholm in order to pick up the prize. And in doing so, we really found out what it was like to be a celebrity. I'd never really had this experience before. And, you know, the thought of journalists coming along and wanting to interview you and ask you questions and so on. At first, it was very nice, but then it got a bit wearing, and I wanted to do science. I love to do science. And so I tried to temper my activities. But of course, one of the things that happens, you get loads of invitations to go and do fun things. And in this case, fun things involved a lot of travel. So I got invited to, well, many places, just too many to recount. And my wife and I had 2 young children at the time. My kids were very young, so it was not that easy to travel, but they were old enough that they could enjoy it. And so we went on a number of trips. Then later on, when my kids went to school, then they couldn't go on quite as many trips, so I did quite a lot of traveling separately. But one of the things that happened, I made a discovery, which I probably should have realized right from the start. And that is when you win a Nobel Prize, all of a sudden people start listening to you. Previously, I would talk about things that I felt strongly about, particularly a lot of examples of the misuse of science or humanitarian causes that I was very in favor of supporting. And I talked about these and nobody really listened. But once I won a Nobel Prize, people started to listen. And there were several things that led me to continue. One of which was to really push for open access in the literature. And this was really the first Nobel campaign I ran, to do this, to get open access in the literature, to try to shouldn't have to pay in order to read the research that had been done. Another one was Libya. I helped get a bunch of Bulgarian nurses and one Palestinian medical student out of prison in Libya. And that gave me really a tremendous feeling, tremendous inspiration that I might be able to do something. And again, all because of this lucky discovery that led to the Nobel Prize. One discovery that we did make with Xiaodong Cheng, who is a very talented Chinese scientist. He was at Cold Spring Harbor at the time. He's now down in Baylor College of Medicine. We discovered that when DNA methylases bind to DNA, they flip the base. Shown here, the bases in DNA are shown in yellow. You can see the one on the right that is flipped right out of the double helix and into the protein where the chemistry gets done. This turns out to be a very common mechanism for all sorts of enzymes that do chemistry on DNA. And this was discovered just a few months before I had to go to Stockholm to pick up the Nobel Prize for discovering splicing. And so, because I'd not worked on splicing or any of the follow-up things for so many years, I actually made this the focus of my talk. And, you know, if you make a discovery, it's always good to be able to tell the Swedes about it. Now, before closing, I want to do a couple of things. The first, though, is to really talk about the importance of luck. I mentioned this right at the start. I learned this in a very practical way when I was playing billiards. When I was 17, I stopped going to school because I got bored, and I used to go and check in the morning and then go and play billiards all day, and then go and check in the afternoon so they'd think I'd been there all day. One of the teachers spotted this, and I thought I was going to be thrown out of school. And the thing I did best was I played billiards. I was a very good billiards player, a good snooker player. I was West of England junior champion. And one day I was playing in a tournament and I made a lucky shot. I was trying to do one thing and it didn't work. And instead, I potted a ball that I wasn't expecting to pot. And then the next shot that I made after that, as I was trying to continue the break, I fluffed it. It just didn't do it properly. And afterwards, an old man came up to me. He must have been in his 80s, but he was a very, very nice man. He came up to me and he said, you know, I watched you play today. You're very good at billiards, but if you're going to be really good, one of the things you have to learn, not only for billiards, but also in life, is that when you have a lucky break, you have to concentrate sure that you take advantage of the luck. Because if you don't take advantage of the luck, what was the point in having it? Everybody has luck. Most people take advantage of it. Why shouldn't you? And that's something that stayed with me throughout my career. Very, very important to take advantage of luck. And so Kazu Kurosawa, this Japanese postdoc when I was doing chemistry, who taught me so much about chemistry, he was one of my lucky breaks. And I actually met him very recently in Japan, and met him and his children and even his grandchildren, and we had a wonderful reunion. And I was able to tell him just how much I appreciated everything that he'd done for me when I was a student, a graduate student, and he was a postdoc. Another piece of luck was going to Harvard instead of Wisconsin. Wisconsin would have been good. I'm sure I would have had a good time there, but I would not have met the large number of people that I met as a result of going to Harvard, and many of whom have become very good friends. Another example, looking for eukaryotic promoters in adenovirus. We were just looking to sequence a promoter. We weren't looking for anything special. Two things came out of that. One was that we discovered there was a protein covalently bound at the end of adenovirus DNA, which turned out to be one of the first examples of such things, of proteins covalently bound at the end of linear DNAs, and they're expected to be there now because they help replication. But it was really looking for the promoter, failing, and then discovering RNA splicing. And I learned several things there. One was the importance of luck, but the other was the importance of failure. And so when students come to me and said, oh, you know, I did this experiment you suggested and it didn't work. And I always say, oh, yes, that's interesting. Tell me all about it. Because my interpretation is usually not, well, the student messed up, but that nature is trying to tell us something, that what we thought was true when we designed the experiment really isn't true, and something else is going on. And so I think it's very important, certainly for anybody doing research and having students, to teach the students that failure is a good thing, but you need to do a post-mortem. You need to find out why you failed. Sometimes it is just because you spat in the tube or you dropped the tube on the floor and picked it up again. But sometimes it's because nature is trying to tell you something. One of the very luckiest things that happened to me was taking an early plane. You probably remember in 2001, there was a terrorist plane that went into the World Trade Tower in New York. I was booked on that flight until 2 weeks before it took off. I was going to a meeting in California, and the meeting got moved 1 day earlier. And so I ended up taking that plane the day before it went into the World Trade Tower. Now that is luck. Had they not moved the meeting, I would not be with you today. Another thing, studying bacterial RM systems. This was a lucky break. It happened back in 1972 when I first went to Cold Spring Harbor. And I've been so fortunate that many, many good discoveries, nice papers, wonderful moments of discovery have come from studying bacterial RM systems. Also led to New England Biolabs and the importance of New England Biolabs, the importance of entrepreneurship, which I think is something that is also very valuable in Africa. In fact, we're thinking of setting up a small company in Africa that will emulate the start of New England Biolabs. The idea is to find an entrepreneur who would be interested in doing something and would be interested in setting up a company. Enough money that you could support research, do some research within the company in much the way that we set up New England Biolabs. As I say, when I first joined them, there were 3 people. We are now a company of 650 employees, and we have more than all paid for by the company, don't have to write grants, and we're planning on increasing that number, doubling it, in fact, over the course of the next few years. So there are many ways to support research, and I know in Africa in particular, it's always a problem. But if you have a company and you make money, then you can use the profits to support research. And I think that's not a bad thing to aim for. I just want to talk very briefly about 2 post-Nobel activities. I mentioned already getting the nurses out of Libya. I started a few years ago a pro-GMO campaign. The GMO technology was developed a long time ago by a man called Mark Van Montague and colleagues in Brussels. And it was immediately obvious from the work that he was doing, and Mark had been a very good friend of mine for many years, it was obvious from the work he was doing that this was a way to improve breeding crops. And so he went on and showed how one might be able to do this. A number of companies, including Monsanto, picked up on this. A lot of genetically modified organisms have been made it out into the field. BT corn, which is now grown widely in the US. It's also grown in South Africa. Many other things have come about. And it's clear that this is a much better way of breeding plants using genetic modification techniques than the old method. You can do it quickly. You can. In Nigeria recently, the cowpea plants have been genetically engineered. They do much better. Cotton has been improved so that it does much better. And it is unfortunate that a group called Greenpeace in Europe decided that this was dangerous, and they have made political hay and caused immense chaos around the world by being opposed to it. And I have been working with my Nobel laureates to try to convince both Europeans and governments in the rest of the world that GMOs are safe. Everything we know about them says they're safe. As a means of breeding crops, they're much better than traditional breeding methods because they're faster and they're more precise. And if anything, the traditional breeding methods are much more dangerous than the GMO approaches. And so this is something I feel very strongly about. I will continue to support this. So far, we have 159 Nobel laureates who've signed on to this, and we're seeking more. So I started a campaign together with Roald Hoffmann, who is a chemist. He developed the Woodward-Hoffmann rules, won the Nobel Prize in 1981. And we are trying to do something to support Ukraine, to let scientists in Ukraine know that they're not alone, that they're supported by us. That going after Ukraine and starting this war was a huge mistake and is not something that they should ever have done, and they should get out of it as soon as they can. Within 2 days of asking, 166 point, we should make it public, make the campaign public. If you go to this website, www.nlcampaigns.org/Ukraine.html, you will find the letter supporting this. You will find the names of all of the Nobel laureates. This will be updated pretty much every hour if more people sign on. The campaign appeal letter is in Russian, it's in Ukrainian, and there are also letters of support from the Dalai Lama, from Jimmy Carter, and from President Obama, all decrying what Russia has been doing in Ukraine. And so with that, I will stop. Happy to answer any questions. All of these slides will be available on my website, and I'm sure very easy for you to find.
Wow.
Not all in your way. Thank you.
Ladies and gentlemen, I think he deserves a standing ovation because he's giving us a piece, a semblance, an image of what happens in the Nobel Hues in Stockholm. So another round of applause for Sir Richard. Thank you very much, sir, for being so kind and gracious and humble. Every time I listen to Sir Richard, he talks about the elements of luck. He does not claim everything for himself. He acknowledges the people who have contributed to who he has become and is very, very humble about his achievements. So I would like to thank you once again for accepting to do this. Please permit me to do some introductions by name, since these were specially invited to be here. The Pro-Vice Chancellor obviously has been introduced, Professor Lisowusu-Dabo. The Director of OGL has been introduced, Professor Philip Ncheje. The Vice Dean of the School of Graduate Studies, Professor Michael Poku-Buensi is here. Professor Leonard Amekuji, Provost of College of Science, and my Provost is here. Professor Makaduma Samoa is being represented here, the Provost of Engineering. And the University Librarian is being represented here by Mr. Michael Ahenkrama, who is the Deputy Librarian in charge of research. We have the media here and the YFS team who will be introduced later. Wafa Adujemfi, the Graduate Students Association of Ghana president, is here, GRASAG. And joining us, and then also Dr. Jacob Hamidu, Animal Sciences here, a friend of the Young Researchers Forum. Another person from the library, Mr. Yao Aviamu, is here. And Dr. Serebuati from the Department of Physics is also here. Reverend Sister Professor Eugenia Ampofo, the Dean of the Faculty of Social Sciences, is here. Professor Christian Ejare, my friend and mentor, the Provost College of Health Sciences, is here. Thank you very much, Prof. And then joining us virtually, we have Professor Charles Marfu, the Provost of the College of Humanities and Social Sciences, and Professor James Hawkins Ephraim, former Dean of Graduate Studies and mentor to a lot of people here, including myself, is also joining us virtually. Thank you. I'll hand over to Dr. Lena Mensah, who will be, in case we have any chats or introductions in the chat that she would want to pick up. Any introductions in the chat? Nothing. Okay, we thank you very much. Virtual audience, it's now time for questions. And I will start the question. Sir Richard, I would like to find out how the message, the delivery of the award or the nomination or selection of the Nobel Prize was given to you. How was the news delivered? What were you doing at the time? And how did you feel?
Okay, so I heard about it actually from a reporter from CNN. So the Nobel Committee had tried to reach me, but they had the wrong telephone number. Unfortunately, I get up pretty early most days, and I used to get up even earlier. I was up about 6 o'clock or so, and I was working on a manuscript, working on a paper, and the phone rang. And I was actually pretty annoyed for the reason that my wife was still asleep upstairs, and the telephone was bound to wake her. And I thought, well, you know, I'm going to be stopped from doing this work. So I went to the phone and I was, "Is this? Why are you calling at this time in the morning?" And the voice at the other end says, "This is CNN. Have you seen the news?" And I said, "No, why would I be watching the news at this time in the morning?" And the voice of the Nobel Prize. And so after that, it didn't seem quite so bad. So I didn't hear from the Nobel Assembly until about 11 o'clock when they managed to get through to me. But in the meantime, of course, my phone had been ringing off the hook from people who'd been calling in to congratulate me.
Thank you very much for sharing that story with us. Any other questions, please, audience?
Yeah, I'm Alexander Yadebra, a member of the Young Research Forum. So thank you very much for the impressive talk. And you kept on saying luck, 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 luck. And I could see you work with a number of great scientists, including Nobel Prize winners. So no wonder you became one of them. It's worrying that so far there's no Nobel laureate from Africa. And what do you think it would take to get a Nobel Prize winner from Africa? Thank you.
Yeah, you're really talking about science. So I don't believe there are any scientists working in Africa who've won, but there've been
Yes,
Peace Prize winners.
the Peace
Plenty
Prize.
of Peace Prize. Yeah, right. So in fact, the year that I won it, Nelson Mandela won the Peace Prize. And I was fortunate enough to meet him because he and de Klerk, you know, the Peace Prize gets given in Norway. They had business in Stockholm. And so we had a meeting with them at the Nobel Foundation. And I ended up having about half an hour talking with Nelson Mandela, who is probably the most inspiring person that I've ever come across. He was just fantastic. Never met anybody quite like him.
Yeah, so sir, the question is, how do Africans become,
Right.
what would be your advice to an African who wants to become a Nobel Prize laureate in the future?
Well, the first thing I would say is it's always a mistake to make becoming a Nobel Prize winner your ambition, because there are so few that are ever given that the chance of achieving it are very small. But to do good research, this is an excellent ambition, and this is something that you should try for. And I think the way to do it in Africa is to get the very best education you can while you're young, and then to go abroad in order to go to university and continue your studies in university. But then when you're finished and you've learned, go back to Africa and see what you can do to help Africa. And this is one of the reasons why we want to start this small company in Africa that will, we hope, generate some money for research, and will show how it is in fact possible to succeed in Africa, to make some money, to do research. We anticipate the company will be making research reagents, things that we would normally make here and then ship to Africa. And we hope that if the company is successful, that they will head off in different directions and do some things that we would never have thought to do ourselves. Because, you know, Africans, I have the greatest respect for them. They are very, very clever people. They're driven. Some of the smartest people I've ever met are in Africa. And they would have been world famous, except for the fact that it is difficult working in Africa. It's not easy. I recognize that. But you do have to find a better way of electing leaders in many cases. Unfortunately, the level of corruption in Africa tends to be quite high. And I must say, I would offer as an example, President Zelensky in Ukraine, who is a comedian. And perhaps this is what you need. Perhaps this is what we all need, is to really find comedians who can lead our countries.
Thank you very much. I hear those joining us virtually, there are some hands up. Please type your questions in the chat so that we can read them out. Any other questions?
Thank you, Professor Nkansa, for coordinating this. Sir Robert, thank you very much. I've been using your whatever you have discovered to be teaching, so I never imagined meeting you today, so I'm also lucky. So I have two questions. One is about the entrepreneurship component and your science discovery. I realized that you were able to move your discoveries eventually to make it a business. As a young scientist, I need advice about how, as part of our own research, we can create businesses or entrepreneurship out of it. Then the second one is about the later part we are campaigning for.
I can't hear anything. Okay, so can someone let me know if you can hear me? Can you raise your hand if you can hear me? Okay, so why don't I take a crack at that first question about how you become an entrepreneur? And I think if you're doing research, you often discover that there are things that you are doing Oh, this is a problem here. Well, now I don't know what's going on at all. Okay, can you still hear me or not? You can, okay. Okay, so let me just briefly deal with this question about being an entrepreneur. When you're doing research, you often have two components to what you do. One is thinking about the experiments, and the other is really where do I get the equipment, the reagents, everything else I need in order to do the experiment. And very often, if you're really innovative, you will be technology and techniques that other people haven't done before. And I think if you can come up with a good technology, something that you discover how to get results from, this is something you can often commercialize. And so always think, what are the problems that I'm facing? Can I solve them? What is the technique that I'm using? Is this something that other people could use? And in this way, you can often make a small company, or join a different company, or persuade a company to pick up on some entrepreneurship. And you don't necessarily have to start it yourself. You can go and join someone else. You can find a businessman who may help you. In the case of the company that we are trying to set up in Africa, we are going to be the partner. We're going to provide the cash to enable a young African scientist to start a company. And if they are successful, they eventually will own the company and we will step back. We're not looking for return of the money or anything like that. We just want to get it started. And I think if we can show that that can be done successfully, there are many other companies that have done well and will want to do the same. So I think almost everybody that I know has a great respect for Africa, and we would like to help scientists in Africa, just as at the moment we would like to help scientists in Ukraine and around the world. Science is very much a world activity. It's something that if you're a scientist, wherever you go, then you have friends in other scientists. Just go to a university, find a scientist, you have a friend. So is there another question that I can answer? Because I can't hear anything from the far end. Oh, here's an interesting question I see in the chat box from Charles Marfo. You ask about religion. I'm an atheist. I could tell you a story about religion. When I was young, my parents, who were not religious at all, sent me to a Sunday school. And so after going there for a little while, I didn't like it. And so they didn't make me go. And I never again found religion until I was 16. YMCA, Young Men's Christian Association, but it was just a club where I learned to play billiards. And I was asked to give a talk to an audience one time in the local church, as sort of a Christmas message. And when I was there, there was a young lady who was in the choir who I thought was very beautiful. And she was quite religious and tried to persuade me I should be religious if I wanted to go out with her. And I actually did that for a while. And then she got rid of me. She said, no, I don't want to go out with you anymore. And so that was the end of religion for me, I'm afraid. I like to believe in things that I can see solid evidence for. And I'm afraid I have not yet seen any solid evidence for God. Let me see if okay, so you talk about the entrepreneurship. So the company that Biolabs is planning on starting, there will just be one. It will be, I will get involved in it, I'm sure, but it's really Biolabs who are going to get involved in it and put up the money for it. And what we're hoping is that it is successful and that it will become an example for others. And I think we may then find that there perhaps will be other Nobel laureates or other companies that would like to do the same thing. But we view what we're trying to do merely as an experiment and hopefully can show that this approach will work. Oh, I see there's also a question here about open access from the TWAS Young Affiliates Network. So I do deal with TWAS from time to time, and I would be happy to support that, and I'm sure we could find a large number of people who would support that. There is definitely a move in the developed world that journals should become open access. Many journals that never were are beginning to think that this is the way to go. And if so, that will be a big help for Africa. But I think this is just something that you should perhaps write a correspondence piece or an editorial or something that could go into Science and Nature, pointing out the importance of this. And I'm sure this is something you could get many Nobel laureates, many businessmen, many scientists to support, because without open access, so much of science just never makes it out to the general public and never makes it out to other scientists. And in particular, also to a lot of companies. They often have trouble accessing the literature because it's very expensive. I'm trying to see if there are any other questions in the chat that I might try to deal with. Right, so here's a question that deals with something that I mentioned about young people changing fields and moving from one discipline to another. I think a lot of the time when you want to move from one field to another, sure that you talk to wherever you're going to go. Let's say you're applying for a postdoc position and you want to change fields completely. Make sure that in your letter, you explain why you want to change fields. And most importantly, explain your passion for the field that you want to move into. Because I think you will find that everybody who really wants to hire students, postdocs, anybody, you're looking for someone who is absolutely passionate about what they want to do. Because people like that work much harder. If they think their job is their hobby, they work at it because what I do is my hobby.
All right. Hello.
Hello.
Can you hear me?
I can.
All right. We have a question in here for you from the provost of the College of Health Sciences.
Thank you very much, Prof, for the moderation. Yes, Robert, first I have two maybe comments and at the same time the question. First, you talked about the entrepreneurship and you demonstrated it in your research work and then especially the EDI phase. You moved to Cool Springs Labs, which is not a typical academic environment. So I want to find out how you manage with the postgraduate training, in terms not only postdocs, but postgraduate training, especially PhD or doctoral training. And again, I know in the US, they are more particular with, based on the categories of funding or grants, targeting research or typical academic institution, the universities. So how were you able to navigate through this? Would you not be in, your institution not being a typical academic institution? The last, you've been a mentee, a mentor, and maybe a grand mentor now. So I want you to share with us some of the tips or the tips that we can as young researchers, and we have some of our colleagues here who are also mentors now, how we can manage so that we can get to the top as you are now. Thank you.
Okay, so let me say a little bit about how to sort of deal with the system. So I was fortunate in that when I'm in England, I got a scholarship to go to university because at that time, everybody who went to university got a scholarship. This really should want to send the brightest young people to university. Whether or not they can afford to pay for it themselves really should not be an issue. So I was fortunate that was paid for. I got a postdoc position at Harvard that was paid for from one of Jack Strominger's grants. And then when I moved to Cold Spring Harbor, I had my own funding, my own grants, eventually. But to start off with, there had been a big block grant from the National Cancer Institute that supported Cold Spring Harbor, and I was hired into one of those positions. And I think you will find that, let me give you one example. There is a young lady working at the Maiden Biolabs who is in our production department. She was born in the US, Nigerian parents. Her parents moved back to Nigeria with her when she was very young. She grew up in Nigeria, got educated in Nigeria, but then realized that she was not going to have the opportunity for a university education if she stayed in Nigeria. So she moved to the US by herself, got a university education, decided after that she needed a job. She applied to us, we took her on. She is now interested in pursuing a PhD, and I'm trying to introduce her to people. She's passionate about immunology, to people at Harvard University who know about immunology and would be interested in taking on her as a graduate student. We as a company typically pay people who work for us and help support them when they go and seek a university education. So we actually make a point of helping our employees to seek further education. And I think there are a few companies that do the same, but maybe not as... And this is an issue where it's sort of company philosophy. This idea from companies who look for nothing but to make as much money as possible for their shareholders. I just don't like that. And I think it doesn't benefit a country. I don't think in the long run it even benefits the people who are simply making money from this. How much money do you need? You don't need a lot of money in order to have a happy life. And if you have extra, then use it to support students. This is such a good thing, very good thing to do. And now you also asked a question about mentoring and what you do. And I think what students are passionate about, what it is that they really love to do, and hopefully things that they're good at, hopefully they like to do things they're good at, and then to make sure that they have every opportunity to do that. Don't try and force them to do one thing or the other that you think is good. Find out what it is they're passionate about, or inspire them to be passionate about something. Because, as I say, if you're passionate about what you do every day, you go to work on a Monday and on Friday, they say, what am I going to do over the weekend until I can go back to work on Monday? These are the kind of people that you want working for you. And I think if you look around, there's no shortage of them, especially in Africa. I've found so many African students who are just really keen to work and get on and to do well. And I think let them go abroad, encourage them to go abroad, get an education if they cannot get it in their home country, but then come back. Make sure that they come back. This has worked very well for Singapore. Singapore has a very good program. They take the brightest Singapore, and if they don't go back, they have to pay for their education.
Thank you very much. Prof. Chair, please permit me to take one more question.
Thank you very much, Sir Roberts. I've really, thanks.
One moment, please. First of all, the official formal way of addressing me would be Sir Richard.
Sir Richard.
I would much prefer that you just call me Rich. All my friends call me Rich, and I'm sure if we met, we would be friends.
Okay. Thank you very much for your presentation, Rich. I would like to move you a little bit from the physical science to social science. I am not in the physical science, but I'm in the social science. And I know that we have Nobel laureates in economics as well. And when you listed your lacks, one of them was that you had access to the right network that helped you to advance in your research. Are you aware of any such network for those in the social sciences like economics, the laureates network that we could link up with and get some mentorship or get some kind of information from them with regard to how to advance in our own area?
Okay, so I don't know the answer to that. I don't know if there is such a network, but I suggest you write to me
My email address is roberts, R-O-B-E-R-T-S, .com. Write to me, ask me that question, and I will pass it on to a number of my economist friends. I know a number of the Nobel laureate economists, and see what they suggest, and either ask them to write back to you and give you an answer, or write to me and I will convey the answer to you.
All right, thank you very much. And on this note, I'd like to say a very big thank you to you, Rich, for your time, and also to all of you audience for being so charming. Another round of applause. We'll now take the closing remarks from the Pro Vice Chancellor who chaired this function for us.
Thank you so very much, Professor Marian Nkansa. And Sir Richard, my disappointment of you is in the fact that in all your achievements and with your persistent character and attribute, you failed to pursue religion because a lady failed you. But there you go. You don't win them all, do you? Sometimes, it's you're successful in the lab. You do all that you have to do, and you come up with all the great achievements and great discoveries, and yet you find out that some things are elusive, and you never can catch up with those. It tells you about life and the reality of life. Colleagues, what an amazing way to end this. For me, we could go on and on and on and on, and listen to you, Rich, and listen to all these stories that you have to tell us, all the way from life in England, moving to New England, and the lessons, the spicy nature of the lessons you've learned, the many lives' true stories that you've shared with us today. Pre-Nobel laureate, during Nobel laureate, and of course post-Nobel laureate. Your environment in Bath reminds me of the environment there. It's such a beautiful scenery in Bath, and I'm sure that you do go there as often as you can to visit folks there in Bath. And I also schooled in Nottingham, close to Derbyshire, so obviously I understand the hills and greenery of Derbyshire, and hopefully you do tend to go there and look at your school where you had your basic education. But importantly for me today, you have shared very important lessons of perseverance, of even sometimes getting negative results, and how sometimes negative results can translate into positive outcomes in life, and how sometimes a perceived failure were translated into tangible research outputs, which has transformed our understanding of biochemistry and certainly the role of enzymes in all the work that we do. I am also thrilled by your mentorship, the fact that you yourself were a mentee of Watson and others, Krebs and so on, and all of those persons informed and influenced and formed your character and attributes in both the laboratory and in your personal life. And hopefully we gather here and those online will pick some of these important attributes as mentors, as scientists, as individuals who are in pursuit of knowledge, and will create the necessary environment for the young ones to also grow their profession and beliefs, so that together we can create that necessary research citizenship that we so much crave for. And one big lesson which you taught or mentioned today was in the fact that we need to believe in Africa. And I'm sure that all of us seated here have tasted of life in the West and perhaps in the East, and we're back home. That's only because we believe in Ghana. We believe in Africa. And therefore, we are setting in motion the processes that will translate into very important tangible outcomes. And also in the belief of knowledge and the pursuit of science and the tangibilities thereof. I want to thank all of you virtually and in person here for your presence and for supporting this noble cause of definitely engaging with renowned scholars from far and near. And today we have been delighted to host Sir Richard, who prefers to be called Rich. And why not? He is truly rich in the sciences and the pursuit of knowledge. Thank you so very much for coming. Thank you.
Thank you very much, Prof, for doing this for us. I'll now invite Dr. Edmond Kwaji to give us a vote of thanks.
Thank you, Prof. Nkansa. So, Prof VC did the thank yous for me, so I would try and keep mine shorter. So, we are grateful to you, Sir Richard Roberts, for your gift of time and kindness towards us today. We cannot thank you enough. For university management, Pro-Vice Chancellor, Provosts, Deans, heads of department, we acknowledge your interest and support towards young researchers and the Young Researchers Forum. And hopefully we would reach that destination where we have a critical mass of research citizens. For our colleagues who have joined us online, we thank you, and we thank you that you were able to stay connected and listen to this very first session of our Research Scholars Dialogue. We can only say that let's ride our luck together, and we would again get there. Thank you all very much for making it possible to be here for this maiden edition, and we look forward to seeing you again the next time round. Thank you, and bye from the Young Researchers Forum.
So, ladies and gentlemen, this is how we end the maiden edition of the Renowned Scholars Dialogue of the Young Researchers Forum. Thank you so much for joining us. See you next time. Bye-bye.